So good morning, good afternoon, everybody, and welcome to the third and last webinar of the VALITEST webinar series on the use and validation of high-throughput sequencing tests for diagnostic of plant pests. So for the one joining for the first time, VALITEST is an EU-funded project undertaken by a consortium of 16 partners and built to improve the reliability of diagnostic tests performed in plant health laboratories across the European and Mediterranean region. And you can find more information on our website on the project. You have the link in the chat. So the objective of this webinar series is to present the recommendation developed by the VALITEST Web Package 2 on the process of selection, development, validation and verification of high-throughput sequencing tests, and the quality assurance of their routine use as diagnostic tests. So these recommendations are general to enable a broad application in plant health fields with appropriate flexibility to account for the changes of technologies. So after the introductory webinar on HTS and the webinar on how to prepare your laboratory to conduct HTS tests, today's webinar will focus on how to develop, validate, and routinely use HTS for diagnostic purpose. So the presentation will be recorded, and a link to the recording and to the PDF presentation will be provided on the VALITEST website. And also know that some polls will be asked, not in the GoToWebinar platform, but in WooClap, and you will need your mobile phone or a web navigator to answer. So today, again, our speaker is Sébastien Massart. So Sébastien Massart is a professor at the University of Liege. He is a plant pathologist interested in diagnostic improvement, plant virology, and plant microbiome research. He is currently leading a research team of about 10 scientists in plant pathology. And that's it. So I will give the floor to Sébastien for this 1 hour and a half webinar.
Thank you very much, Charlotte. And I must say something to the people who will come to the online training activities. We won't repeat for the 4th time the same introduction, but it's needed because there are new people at each seminar. So thank you, Charlotte. So today, third step, it's about adopting high-throughput sequencing technologies in a laboratory. And I insist in a laboratory because, again, as I told yesterday, but I think it's really important, it's these guidelines and what I will explain today is important for and is essential for a diagnostic laboratory, but it's really good to think about and might be nice to go as far as possible for a research laboratory. So what I would like to ask you also throughout the session, have in mind or write on a paper, whatever you want, in fact, keep in mind these two aspects and think about whether you are a researcher or you are a diagnostician, but with a former researcher past. Think about all the steps and all what I will describe today. Which one of them could be mandatory or could be recommended for a research lab? What is like the minimal level of evaluation, validation controls that would be required for a research lab? So think about it throughout this presentation, because at the end, I will ask you this question, and we will see a little bit how collectively we have ideas on that, and if we have some common basis that might be shared between each other. Okay, so applying high-throughput sequencing in the laboratory, and the guidelines, in fact, have been written thinking on 3 applications. Either applying the shotgun sequencing, remember library preparation shotgun, on a mixed sample. What does it mean, mixed sample? We consider, for example, a single plant as a mixed sample because you have several organisms within it. Yes, there is the plant, but inside the plant you can have bacteria, you can have fungi, you can have viruses, and so on. So we consider it as a mix. We can also apply within the guidelines the amplicon sequencing from a mixed, again, a mix of spore, a mix of plants, a mix of insects, whatever it is, making a PCR targeting a region, allowing taxonomical identification and analyzing the results. And the third possibility that is envisioned in the guidelines, this is the shotgun sequencing from a pure culture. In that case, it's a bacteria or a fungi culturable on petri dishes, which is a pure culture. You take you make a genome sequence from a single colony forming unit. So that's the three kind of application that we have to think about when designing and writing the guidelines. The guidelines, as I will explain today, and applying them in a laboratory, there are these 6 different steps. We will review them progressively today. And what we have also included, it's a risk analysis, which obviously is spanning all those different steps and all these different elements to be applied. And in addition, what we will also discuss today about the controls that are also important throughout the selection, development, validation, up to routine use of high throughput sequencing technologies. Okay, so first step, selection of a high throughput sequencing test. I already have a word on this yesterday, that in fact you must define very well the scope, which best metrics you will target. And these tests should be selected depending on this, but also all the practical constraints that there are in the laboratory, but also the availability of validated official reference tests. Anyway, the selection, and as I really insist from Friday on this, it's both for laboratory, but also for the bioinformatic part, which is a really important step in high throughput sequencing. So let's go for a first practical evaluation altogether and to see a little bit what you think about which high throughput sequencing test would you use in several situations. So you can go here on the website. This is the same as yesterday and as Friday. Just you scan this QR code. If you are in a browser, just write this this address, and I will start a pool there. Here it is. So the question is pretty simple, and I will put it in half the page so you can still have the QR code. Which protocol would you use to detect plant virus in a plant sample? What do you have in mind? If you want to detect plant virus in a plant sample, which protocol would you apply for high throughput sequencing? So your first response, double-stranded RNA. RNA-seq with RNA depletion library, total RNA, yeah. Okay, good. The responses are coming. You still have 10 seconds to answer them. Okay, the pool is over. Clearly, I think there is like a complete unanimity on the fact that we have to use RNA. And indeed, if you want to make a viral indexing, RNA is quite nice because even RNA sequencing allows you to detect DNA sequencing. The opposite is not true at all. And what we can see here, there are specific differences. And I touched on it yesterday while looking on the different protocol existing for plant viruses. And we have it here. We have an option of double-stranded RNA, as you can see here. There is a small RNA, which is also a very popular one. sRNA is the same as small RNA. You have the total RNA, and more specifically, it's total RNA plus ribosomal RNA depletion. And that's a very important point. You must always deplete your RNA, because whether you will just sequence the rRNA from your sample. The important point is that indeed, at this stage, you have selected a HTS protocol, and often you are struggling Should I use small RNA or total RNA with ribodepletion? Which one is the best for my plants, and so on? Not so easy. Just to give an example, for us on banana, we made tests. We evaluate small RNA and total RNA, and we observed that total RNA was actually better to detect the viruses present. But for example, if you have a look on potato, for example, where the silencing is very intense for the viruses, small RNA is performing particularly well also. So again, it's not the different protocol. You should design and think about it in the frame of the plant you need to analyze. Which kind of matrix should I analyze? And another point, it is mentioned here, double-stranded RNA. There is another protocol that is possible also, which is the virion-associated nucleic acid, where you go through an ultracentrifugation to enrich in viral particle. One thing that you have to keep in mind, also a tips for when you want to adopt it, the more steps you will have in your sample, the higher the risk of contamination of error. So even if sometimes, and even in most of the case, double-stranded RNA or VANA will purify your virus and so may improve the analytical sensitivity of your test, you always have much more steps and so a higher risk of error or contamination. But that, in fact, here we have a nice overview of the protocol available to detect a virus in a plant sample. And once you decide for a small RNA or ribodepleted total RNA, think also in mind that there is plenty of the small details that you have to take into account. Which exactly library preparation protocol? There are various providers and various kits for a single provider. What will be the sequencing length? What would be the number of sequence per sample, and so on. So that's only the first step here, but after there is plenty of details to take into account. Second question, if you want to detect bacteria or fungi in a plant sample, which protocol would you use? So I let also you suggest, think about what could be used there, what could be useful to detect bacteria and fungi in plant tissue or from a plant tissue. Okay, by the way, if you want to like what is written, you can also like these proposals. Good, we see there is a large variability, and that will be interesting to discuss also. 10 seconds left. Good, the vote is fixed. So interesting here, because we see from the beginning that we have much more possibilities that exist. If I can make big groups for the possibilities that you have proposed, it's first sequencing DNA. You see total DNA here. You have sequencing the RNA, total RNA, for example, here or here. And the third one is targeted amplicon by 16S, ITS, and so on. Good. In fact, all these protocols are able to detect bacteria and fungi in your sample. The key element is to think about what is my specificity of detection. Can I detect the pest that I look for? And also, what is my analytical sensitivity? Because if you're starting from a plant sample and you just sequence it either by DNA sequencing or total RNA or whatever you do, what you will get actually is plant DNA or plant RNA. Your microbial nucleic acid will be in very low proportion. So it might be complicated to have a good analytical sensitivity there if you want to detect bacteria or fungi in such plant sample. And for example, on total RNA, like here, there is currently the Plant Health Bioinformatic Network, a European project, ERANET transnational project. We have looked into hundreds of total RNA sequencing data made for virus discovery if we were able to detect bacteria and fungi. And yes, we are able to detect bacteria and fungi there, even pests that can be important, endemic pests and so on. but we have few information about the limit of detection, and so the analytical sensitivity. The amplicon sequencing is, like put it here, or 16S ITS. It's an option that can be more with a better analytical sensitivity, because we will specifically amplify the region of the target, bacteria, fungi, but we may lack specificity there. We won't be able to detect up to species level. Sometimes it's possible, but not always. Another element that you have to keep in mind if you work with 16S, you must use a clamp to avoid the amplification of chloroplast of mitochondria. Whether you will get 90+% of your sequence that are actually the chloroplasts of the plants. So detecting bacteria and fungi in a plant are not so easy because their level seems lower than what we can observe with viruses, and there is a huge background of plants that is more annoying than for viruses. So third question, if you have fungi or insects that are in a trap, a spore trap, insect trap, whatever the modality, which HTS protocol would you use in that case to detect them currently or in the future? Then feel free to think what could be the protocol in that case. So I see metabar coding, amplicon, which is similar, total DNA. Okay, responses are coming. Good, and it's now finished. So here we have a more homogeneous proposal, which is many people propose metabar coding or amplicon sequencing, either on ITS, like proposed here, or COI, the cytochrome oxidase, which is a barcode from many kind of living organisms, among which insects. So indeed, it's a good way to analyze the composition of your trap, because you will target a specific sequence, amplify only this one. Here, you also propose total DNA. Several people propose total DNA or shotgun total DNA. As today, you can say, well, I have a complex mix. I will need a lot of sequence. I will need millions and millions of sequence to achieve a good sensitivity. So as of today, it may be still too expensive for a routine evaluation, but you have to think about what I mentioned Friday. The price of high throughput sequencing is going down and down and down over the year. Yesterday, I showed you the example of €3 per megabase on the NovaSeq 6000. So that means even if today total DNA might be too expensive, it might be an option in the 10 years, I don't know, but it's something which could be possible further on. But clearly, as of today, people are using this metabar coding on COI or ITS to evaluate the composition of fungi or insect from a trap. Here you mentioned ONT, Oxford Nanopore Technologies, they were also mentioned before. That's something you will not find technology specificities in the guidelines, and that's something which we were really careful about, is to have these guidelines that go over the technology. We did not make guidelines for the Illumina sequencing or for the MinION sequencing, because once the technology is outdated, the guideline would have been outdated also, and we wanted to avoid this. So that was for the first pool that we made together. So you see, even for a single situation, you always have different tests, and within a test, there are plenty of details which need to be fixed. Let's go on the second step, the development and optimization. These steps are mandatory if there is no official high throughput sequencing standard protocol. As of today, there is no standard. But if you look at 5 years, then we might have some official protocols. either at national, regional, whatever level. The goal of the development is to go from published HTS tests in the scientific literature to ready for validation tests. That means, in fact, that we have 4 key learning outcomes from that. First, you want to gain the experience with the test as a laboratory. Can I handle it properly? The critical steps, the key parameters, and so on. You want also to define what are the most appropriate controls. And it's a very important point that we will address in the workshop, and I will give a word today also. Third key element for the development, you will establish the quality matrix thresholds. What is the minimum number of sequence that I need for the sample? What is the minimum e-value that I will take into account for a blast? What is the minimal length of a context? You have plenty of quality metrics that you need to establish and their acceptable range. And finally, it is also helpful to have a detailed protocol specifying clearly each of the operation of the HTS test. So here, with all this information, you're ready for the validation further on. For the development and optimization, I would like to underline 2 key elements. You decide to evaluate and to develop an HTS test, you need to define a sample panel. On which sample panel I will do it. And you need to define it quantitatively, how many sample, and also qualitatively, which kind of sample I will use. So the guidelines are addressing the elements of reference material. What is a reference material? What do I need as a request for reference material? One very important point for the sample panel, but later on for the control too, is the representativeness of the targets. If you aim to detect viruses in, I don't know, potato, the sample panel must include a representative of the diversity of viruses that you can find in potato. Diversity in terms of type of nucleic acid, DNA, positive single-stranded RNA, and so on. Diversity in terms of localization, phloem restricted or not. Diversity in terms of transmission, etc., etc. So you need to have a pool of target of pests that are representative of what you find, what you want to find. The same for forest fungal pathogen. You need to have reference material, including several forest pathogen, and the ones that will be important later on once you apply just your step within the scope that you have defined. And also the concentration is very important. Very high concentration will allow you to easily detect them, but you will not check for, you will not have previous information on the limit of detection. So it's nice to have also low concentration and high concentration. And remember, with high concentration, there is always the risk of contamination, cross-sample contamination. So the sample panel is important to define properly with a representative set of targets with a range of concentration. And based on that also, something which will be important to define before the validation is the level of pooling that you will do. Remember what I explained yesterday with the index and so on, you pool several samples together. What's the level of pooling that you will do in order to reach enough sequence per sample on your sequencing platform? So that's for the second step. The third element, the third is the validation. So validation, what's happening for the validation? In fact, there are official definitions, and we just repeat them in the guidelines. There is definition according to ISO 9000. It's the confirmation through the provision of objective evidence that the requirements for a specific intended use or application have been fulfilled. We fulfill requirements, basically, providing objective evidence. The ISO 17025, which is really an ISO protocol targeting the diagnostic lab, so particularly useful for an official diagnostic lab. It states that the validation shall be as extensive as is necessary to meet the needs of the given application or field of application. And that's a key element for us. as extensive as is necessary. So that means we have a balance, because if we apply it strictly, I should validate my HTS protocol on all and any viruses infecting potato to be sure I can detect them. But practically it's impossible. So you have a balance between the extension of your validation and number of viruses on which you can apply the validation, and the actual number of viruses infecting this crop. You will never be able to sequence all of them, but you have to think about it, and also to be able to defend your position, your composition, once you have an external audit from in the cycle of accreditation of ISO 17025. On validation, we address in the guidelines some specific points, and the first one is on analytical sensitivity. Here, there is two considerations that I want to share with you on the generated reads, and this is the 150,000 ratio. Let's think we have a theoretical case where a virus is present. You have a sequence of this virus, this viral species, every 50,000 sequence. Very low concentration of the virus. If you generate 100,000 sequence for your sample, you may have more or less 2 reads from your target. So you might miss it, because in some cases you will get no reads at all. But if you generate 100 million reads, you will get 2,000 or more or less reads from this target. So you will most probably detect it. So that's important to have in mind for the analytical sensitivity, and so the number of sequence that you want to generate for your sample. What are the level of concentration of my viruses in my sample? Again, if I go on what we are doing here for indexing banana germplasm, we do not make the test currently on in vitro plants, because the number of viral particles is very, very low. We just grow the plant 6 months in greenhouse in order to get enough viral particle. And so we can have more chance to detect them. The ratio is better. Because on the other side, as I told you, the more you sequence, the better you will see the low abundant virus. But on the other side, I told you yesterday, we have to go from a qualitative to quantitative view of contamination. And the more sequence you will generate, the more background contamination you may have. And this means that, in fact, what we recommend in terms of analytical sensitivity is to determine for the sample a minimal number of sequence to achieve a reasonable analytical sensitivity, which is compatible with the scope of the test. So that's for a key element in the guidelines. The validation also is useful to define the minimal number of sequence. And if you get, for example, less sequence than this minimal number, you resequence it, because you know that if you reach this threshold, you will get a satisfactory analytical sensitivity. And there is also another point with analytical sensitivity, is the presence of multiple targets. We show example in the guidelines, but in fact, once you have several bacteria or several...
Sébastien, I'm sorry to interrupt, but can you confirm that we are still on slide 3? Development optimization of a HTS test? Okay, so it's not changing.
Okay, because I am on the 4th. I will stop sharing and I will reshare again.
Yeah.
Is it... Do you see something?
No.
Boom.
Yeah, now it's okay.
Okay. Okay, so, but there is a delay. Okay. Okay, I see. I will put the panel just near me so I can follow if I get the proper slide. Okay, good. So you see it.
Yeah, now it works. Thanks.
Okay, perfect. So the different targets are competing inside the plants. If you have a fungi that is at very low level in the community, its odds to be amplified might be low. They're competing with each other. And that's something we see also for the plant viruses. For example, a virus alone in a plant will be detected very easily. But if it is in combination with other viruses, it may be a much lower level. So in terms of the analytical sensitivity and the minimal number of sequences that you will propose here needs to take into account the problematic of multiple targets, which means that your reference sample should also be mixes, include mixes of targets. And also, as I mentioned, this low prevalence fungi is competing for reagents. But if you do, for example, an amplicon sequencing and you use a non-template control, meaning you use water and as a blank, you amplify and you sequence it, you will always have sequence from this sample. With very, very low amount of bacteria or fungi, spores that are present in your sample. Very low amount, teeny amount, a few reads, but they are there. And in fact, what's happening is that if you try to look for these targets in your real sample, you will not detect it. Because they're in your non-template control, they are alone. They have all the reagents for themselves, this teeny amount of contamination. Whereas once they are in the sample, just the targets present in the sample will take all the reagents and you might not see the contamination. This, well, very, very low background of contamination. Okay, so the analytical specificity is a second element that was taken into account during that is taken into account into the guidelines. 4 elements to think about analytical specificity. First, what is important, what is the targeted pest and the taxonomy of these pests. How well the species boundaries are defined and how well the different isolates are easily assigned to a species or another. And the question is also brings another level. If you are detecting whole genome and characterizing whole genome, or if you are characterizing only targeted region. And on the other hand, currently, as of today, for example, the bacterial taxonomy, the bacterial genomic is much better resolved than the fungal genomic. So it's much more difficult to assign fungal sequences, for example, from a whole genome, to a species specifically, because the database are not well completed so far. The situation is a bit different for bacteria. And for the targeted region, it really depends on the ability to distinguish different species by the 16S, ITS, or COI. The read length and number is also particularly important because the more read you have, the longer the reads, the better you will have the genome coverage. And the length of the targeted region for amplicon sequencing is very important. That's why there is currently a lot of research on the nanopore technology, because the reads are longer and may allow very good taxonomic differentiation. Big problem as of today, it's not yet accurate enough. So you will introduce too much errors, which could impede or bias the detection of the targeted species and so a proper analytical specificity. Software and parameter, it's about the step 2 of bioinformatics, as I mentioned yesterday, identification of targets. It's about percentage of identity between your generated sequence and the database, the score, and so on. And lastly, the reference database. I mentioned it yesterday with this balance between the completeness of the database and the presence of what I call garbage, which means misassigned or poorly annotated sequences. The validation is also viewed in the guidelines through the selectivity. So meaning, what if I change the matrix? What if my sample is not exactly the same? And there is one very important control, which is the internal control, which will allow to control the selectivity. There is also the repeatability and reproducibility. There we do not propose something else than the current EPPO standard. One of the questions, nevertheless, is how should we adapt this to the specificity of high throughput sequencing test? And in fact, it will be one focus for the workshop of Wednesday and Friday among the restricted number of participants, sorry. But as it is an interactive workshop with small group, we could not accept everybody in the workshop. What we will do, we'll make brainstorming, small group, thinking about cases for validation, cases for controls, and so on, in order for the participant to learn, but also to build themselves and to contrast between each other their knowledge and their expertise in the field. Nevertheless, validation of high throughput sequencing test is, as of today, quite heavy to carry out. We have just finished it for the banana germplasm viral indexing, which means we have had to sequence actually much more than 100 accessions by total ribodepleted sequencing with all the controls per batch, analyzing the repeatability, the reproducibility, making dilution series to evaluate the analytical sensitivity and to compare it with the current indexing protocol and to compare it with what is actually ongoing. Doing this is fine, but you will find discrepancies between both results, both tests. So you need afterward to dig what's happening. Do I have a problem of sensitivity? We discussed yesterday about the sensitivity between PCR and high throughput sequencing. Or is it because it's a divergent variant that is not amplified by the PCR test, and so on. So it raises a lot of questions, by the way, in addition to purely analyzing the data in diagnostic point of view. So
Sébastien, we cannot hear you. I don't know if you are still here. Yes, it seems that Sébastien got disconnected. I'm really sorry, everybody. So we will have to wait for him to be reconnected to the webinar, which I hope will be as soon as possible. Sorry about this.
hello?
Yeah, I can hear you now, Sébastien.
Okay, it seems I have a problem of connection. I go to the office to have the high throughput network of the university and it's crashed. Okay, I think it's okay. No, you hear me and we are the appropriate slide. Okay.
Yeah.
Good. Okay, so verification, in fact, what I was telling, it's when an existing standard validated test exists. Remember I mentioned these tests that are recommended or that are already official standard. For this, you will need to verify this test. And it aims to answer a single question. Can you perform the validated test according to the established performance criteria in the validated available test? So for this, again, there is guidelines in the EPPO standard 798 about the different performance criteria, as you know, and as we explore it. Somehow, and strictly, it's not that, but you can think it's about some kind of light validation to give you an overview. But you need, anyway, you have a validated test, you need to be sure it performs properly in your lab. So you need, anyway, to make replicates to evaluate all these parameters if they behave or they perform in accordance with what is specified in the official test. So that's for the verification. Next is the change management. For the change management, I want to tell you a small story that quite well. In my previous work, I was working for a private company, the Envision, active in high throughput sequencing. And we put a lot of effort in order to be the first or one of the first in Europe to have an ISO 17025 accreditation for exome sequencing service. And as I mentioned yesterday, we had the accreditation just for generating raw data. But it was a big project within the company because we were accredited ISO 17025 from 10 years, but it was the first time we go from classical molecular biology toward high throughput sequencing. And there we worked quite a lot there, analyzing all these performance criteria and demonstrating that we get what we were expected to get using reference sample and so on. So we were very happy to have that. But just after the accreditation, the provider changed the kit. Exome is relying to a kit which enrich in the target sequence the sample before the sequencing. And in fact, these new kits outperform quite a lot and was really better than the previous kit that we had accredited. So we were like in a somehow strange situation because we had an accredited test using an old kit that in fact performed less well than the new kit. And in fact, the shelf life of this accreditation was quite small. It was a nice good learning outcome for that, because in fact the customer, they were aware of this difference. They say, okay, you have the accredited test, but we know that this new kit performs better. We will go for the new kits. So that's something which was very, very frequent at the beginning of high throughput sequencing, but still now there is evolution of technology. So that's why in the guidelines, we have addressed this problem of change management. So the idea there, and I will show you after a flow chart, we need to evaluate the impact of any change in the laboratory or for the bioinformatic pipelines on the reliability of delivered results. And this impact can be either here, okay, this impact is minimal. I just change, I upgrade my reference database and I check on previous data sets, no difference, okay. Could have to do verification, light validation, or I must go for, again, a complete validation. So that's something which, in fact, we propose to think about, and we propose some guidelines to think about. We do not propose solution because every situation will be different. Every laboratory will face different impact potentially of the changes, depending on the technologies, depending on the structure of the lab, the outsourcing or not, and so on. So all these steps before going to the routine use, all these steps are embedded within a risk analysis. And there we had question yesterday on risk analysis. What are the main risks of high throughput sequencing testing? So I have the pleasure today to revert the question, in fact. And I will ask you, again, we will go on the web page from WooClap. I ask you just for you, just go. I have to quit. I will redo it again. Quick. I launch it here. So you can go here again and just, what do you think are the main risks of high throughput sequencing testing? I already told you it's most in the lab, but there is also other risk. I'll just let here sorry, here it is. I put here why we cannot vote now. I open it. Okay, so let's go. You can now vote. What are for you the main risk that you see? Again, I will show here this element. You can like what you found or what your peer has proposed. So I think you are all on the page. Okay. We see risks that are really linked to the sequencing, but also risks that are linked to after the sequencing, for example. And another way to see what you propose here, think also about the risk at any step. Remember these steps I mentioned you, sampling, nucleic acid extraction, library preparation, sequencing. What do you see also as a risk there? There are already some indication, for example, this one, contamination and change in platform. Still 20 seconds to participate. Also, once you agree with a response, please like it so we can see it better. I will put it as a cloud of words. We have contamination in big, which is indeed a big problem. There's still 10 seconds to vote and to like, to propose. If you propose something, no, it will not be liked, but anyway. Still 4 seconds. Okay, so it is now fixed. So contamination, yes, indeed. Contamination is a big risk, and we will see afterwards in the way we have analyzed the risk within the guidelines, that the contamination can occur at several steps. Another here, experience. There was several times the experience about the technical expertise and so on, and that's true. That as there is some specificity in interpretation of results in the, so we can see here, there is a risk for proper, you need proper training, proper expertise of the scientist analyzing or the manager analyzing the results. The low abundance, somehow it's linked with the false negative, or I see also things related to that. It's linked to what I told you. You need to define a minimum number of sequence that allows you to have a proper analytical sensitivity. The regulatory issue is directly linked to the interpretation of results, and we have addressed it in the guidelines indeed. A risk, this is the error in database, clearly. And the other point, for example, sequence is not equal to virus. This is also the same. We could have a sequence which is viral-like, but in fact is an integrated sequence in a eukaryotic genome. I see also black matter in database. Indeed, the black matter, for your information, this is once you generate sequence and you assign them, you have a proportion of the sequence that are completely not assigned. And currently we call it black matter because it doesn't link to any known sequence in database. The validation of the equipment, if it's not validated, is a risk. The risk is also related to the non-adequate controls. And anyway, the way to manage the risk in routine analysis is to pilot it by controls. And we will go on there further on. Too much data and the capacity to, there was also the capacity to handle the data there. So indeed, there are quite, you have addressed a wide range of possible risk. I just want to make another survey. Among you, who knows about Ishikawa and his diagram for risk analysis? I was wondering if he, well, you know it or not. So just have a view on this and let's have a look. Like 20 votes, more or less. So yeah, in majority, you don't know it, which is a good news because you will learn something and you may explain something to your friends or whatsoever tonight or this weekend if you do something with them. Okay, so let's go a little bit, and I will introduce this Ishikawa element. Indeed, Ishikawa is also year of the 6M. It has been done by this for the 6M. And Ishikawa, in fact, is a Japanese engineer in which were very active in the 70s, 80s, and developed a methodology to solve problem. Once you have a problem, think about the material, think about the manpower, think about the what is called the media, well, the environment. Think about the methods that are used. Think about the machine you are using. And so this is the different M of Ishikawa. And that was a way to find the origin of a problem. But you can extend this way of thinking by making a risk analysis. And we use this way of thinking to design a risk analysis for the high throughput sequencing. What is this risk analysis? The Ishikawa diagram is like fish backbone. With you will see we will have a central line here, and we'll have each step will make problems here. We don't do strictly using the 6M methodology, but rather by step. And you will see a brown or blue point, meaning there is a risk of degradation or a risk of contamination. So at sampling, there are different risks that are possible. The matrix, if the matrix change, will it impact my results? The pest distribution in the matrix. When you sample, sampling in spring is not the same as sampling in autumn for some pathogen. The labeling, proper labeling of sampling. We also address risk related to the sample's transport and storage. What could be the recommendation in terms of temperature? Dry highs, blue highs, room temperature, how many times it can be kept at the evening at the -80, the -20, in liquid nitrogen, or whatever. This is linked to the matrix. You go further. Matrix, you will extract the nucleic acid. There, often you make a subsample from your sample. You don't need grams of material. Is there a risk of missing the pest due to this, for example? The homogenization is also very important for a proper subsampling. What is your extraction protocol? Is it well adapted for all your targets, or will it interfere with the proper detection of some targets? And how do you store your nucleic acid after the extraction? If you continue to analyze the risk, you have the extracted nucleic acids, and you have the library preparation step. And we discriminate between the shotgun sequencing and the amplicon sequencing. And there you have some common thing that's in fact the pooling may not be appropriate. The extracted nucleic acid may be of wrong quality, which impede the proper library preparation, or you have contaminant, you have interfering substance. For shotgun, you have the fragmentation, how you shear your nucleic acid. The enrichment, if it exists, the adaptator addition or the amplification can generate bias and maybe a risk of a bias. And here you have also classical risk linked to the PCR cycle. I mean, if you do 40 PCR cycle, you will bias, you will reach plateau phase. And so most probably the proportion of your target will not be representative of the initial proportion in your sample. The sequencing, we also explain some risk linked to the platform, the sequence length, the number of sequence that may impact the result of the analysis. Once you generate the raw sequencing data, you have the first step bioinformatic analysis, and before that, the data storage. Should I have taken care of the integrity, the space, the bytes necessary for the storage? How can I transfer them? Have I secure a backup? For the bioinformatic analysis, is it fine in terms of software, calculation power? What about the version of the software, the parameters used? We have also, from the first step bioinformatic analysis, you have the clean sequencing data, and you do the second step. What about, again, like quite similar to the first step, which software and calculation power is needed, the version, database, threshold, and so on. you get to you identify the pest there, but you also still have to check for the control. And the risk is to have used the not proper control or not the proper threshold or a software versioning. In change management, you change a version of the software. Does it have an impact, for example? And finally, the risk is also linked to what you mentioned, for example, in the cloud, with the regulatory risk, the interpretation risk. When should the confirmatory test be done? What is the expertise required in order to produce, in fact, and at the end, the test report? All this risk analysis needs to be done under quality management program, under quality assurance, quality management system. In fact, in the guidelines, and as mentioned yesterday, there are 28 risks listed. Their cause, their consequences are detailed, and also we propose collectively some mitigation measures to cope with these risks. What is important at this stage, because we review the different steps for applying high throughput sequencing in the lab, but how could we interface them? And what we have drafted is a framework, well, not a framework, sorry, a flow chart, where you have simple question with yes or no. A validated test exists? Yes, just make a risk analysis. Can it be used without modification? Yes, just verify it. And if verified, you can use the test. If you need modification, you need, in that case, minor modification. If yes, you verify. I hope you still have my connection. If there's no minor modification, the modification are major, or there is no validated test, you must do the development optimization, which include, in fact, the risk analysis, and make the validation. And after, you have the test to be used in the lab. That's before using the test, but after, during the routine analysis, you always have the question and the point, do I need an update? And if you need an update, being in the laboratory or in the bioinformatic, in that case, you have to adapt and to redo a little bit your risk analysis. Does it impact my result or not? Is it a minor update? Yes. Does it have an impact on the result? If not, you can go through. If there is an impact of results with minor update, you could verify. And if it's okay, you go back to the test. While if this is not minor update, you change the sequencing platform. For example, you change completely the library preparation methods. In that case, you should go back to validation. So the guidelines aim to guide the labs before doing all the steps before the routine application, but also during the routine application to take into account the change management. So let's go back to one question that will be the last of today. Based on what you've seen, to your view, which steps are mandatory for a diagnostic lab? I will open the pool again. So you go back. You can here simply vote for one or several options among selection, development optimization, validation, verification, risk analysis, change management. What should be applied from the beginning, or preferably what is really recommended and realistic to do for a research laboratory? And I insist on the point of having an equilibrium between resources and extensive tests. Clearly, the best is to do everything. But does it work for a research lab? And I'm curious to see your point of view of that. So you can select several responses. I let the response grow. Okay, let's see, we have 34 responses. Let's have a look on what's your point of view for that. And in fact, what you have in mind mainly, if I see here, it's the development optimization. I will lock it now, sorry. Development optimization is the most important one, and clearly the fact that going from a published test toward a validation ready test is clearly important, even for a research lab on which you apply any HTS test. Validation is clearly important, but really need to be balanced with the available resource for your project. And in fact, validation verification are very nice and important, but somehow you can handle it for a research lab. I insist, for a research lab, through the proper use of control. And you can make like light validation. You evaluate that everything is okay to detect what you have, and let's go on it. So change management seems less important for you. The risk analysis is like quite popular also. And indeed, risk analysis could be nice for laboratory, whatever it is. If I want to use high throughput sequencing for my research on survey in a specific field, whatever, thinking about what are the risks at every step might help you to have reliable result at the end, and doesn't need a lot of resource, because it will allow you after to concentrate your resource on specific points, on specific steps that needs to be really evaluated. And anyway, I want to remind, for a diagnostic lab, all the steps are mandatory, or well, you have to follow the different flow chart in order to raise as much as possible the reliability of your high throughput sequencing test. Okay, so this was for before applying in routine. I will put after in the PDF file, you will have the result of the survey here, so you can see here. So routine application, and the routine application, I will focus on quality checks. And for that, I will just share with you the control landslide. So yeah, let me just go back to what I need. Sorry, here it is. And let's have a look there. Oh, sorry, it's not behaving as was expecting. Just have to go on the appropriate side, sorry. When I make the rehearsal, there was no problem, and now we don't have it properly. Here we are. Sorry. Here we are. I wanted to share with you some thoughts on the control landslide. And I say landslide because somehow we might review a little bit our understanding of the way of making and using controls. So basically, for the controls, you need reference material, and we describe them in the guidelines. We have classical reference material, biological material, or nucleic acids. This is already present in a diagnostic lab. What is pretty new is that you can have data sets as reference material. If you look on the biological material, it can be pest only, a mix of bacteria, fungi, or insects. It can be also a plant infected by this pest, or a plant alone. Think about, you want to monitor the advance of weed, of invasive species. What we really recommend in the guidelines is you use a mix of target for this reference material. I mentioned already it for the development selection and even for the validation, the verification. Use samples that represent a mix of target in order to achieve a representativeness of the diversity of targets. The nucleic acid can be extracted nucleic acid, as for PCR, for example, but you can also use prepared library. I mean, once you prepare a library, you can sequence it again and again and again and again. There's a huge volume of library compared to the tiny amount that we put on the sequencing machine. And for the datasets, I just recommend you to have a look here on the results from IRANET project, a FRESCO project, the Plant Health Bioinformatics Network. coordinated by Annelies Hagemann, where we designed it. In fact, Lucie Tamizier, no, she's a postdoc in Avignon in France. She designed it, well, not alone, but all together, we designed semi-artificial data sets to validate bioinformatic pipelines. And these semi-artificial data sets are aimed to simulate specific challenges for virus detection in terms of analytical sensitivity, of selectivity. And so they might be a useful resource as reference material and as control of the bioinformatic steps. For the control, we are on the control. There are the classical one, positive control. We recommend a mix at low concentration, which allow to evaluate the analytical specificity. Do I detect all the viruses, all the bacteria present in my mix at very low concentration? Sorry, it's for the sensitivity. You can also evaluate the analytical specificity. Do I detect properly the different species that I have put in my sample? And also, and that's also new for that, it can serve as a negative control. Indeed, you have a mix of 3 species, 5 species. Any other species detected in your positive sample might come from a contamination. And so the positive control can act not for all the targets, but at least for the targets that are not in this control as a negative control. You have also the alien control. And this is quite a new control here. It's in fact an alien target. That means that it's a target. So if you look for viruses, it's a virus. You look for fungi, it's a fungi. But this target could not be present in the sample you analyzed. To give you an example on what we are doing here, we use high throughput sequencing to evaluate virus infection of banana germplasm. Our alien control is a sample of wheat infected by BYDV, the barley yellow dwarf virus, which cannot infect banana, in fact. And so I use it as an alien control. It's a sample, an external control. And it can check for contamination. In fact, the contamination, what I can do, if I have one alien sample, I could check in all my other sample if I can detect BYDV. And if I have BYDV in my banana sample, that means it is contamination. And on the other hand, from my sample, I can evaluate if I get, if in my alien, I can also evaluate if I have target for my sample. Do I detect banana viruses in my wheat sample? So this is a way to evaluate, and at the end to evaluate quantitatively the contamination between the sample. So the alien control was not proposed so far, but will be most probably a very important player in the reliability and the contamination evaluation in high throughput sequencing. You mentioned contamination as a very important element. This alien control can help you to identify contamination. There is also the internal control within each sample. For this, it's a sequence from an host plant or an alien target spiked at low abundance. And I mentioned the publication of Mike Rutt and his team in Canada, where they use viruses, they spike it at very low concentration. And it's like the alien. It's a virus that cannot infect the plant that you are analyzing, and you put it at low concentration in the sample so you can validate the results. If I detect properly my alien internal control, I'm okay. I achieved the proper analytical sensitivity. And so it can validate each sample and the result of each sample to be sure I don't have a specific inhibition or whatsoever in this sample. And finally, and I put a question mark, the negative control.
Sébastien, I think we are stuck again.
Yeah, I see. It's really annoying. I'm really sorry. And I am on direct. I'm on direct network, not Wi-Fi. No. I restart my connection. I stop my. Do you hear me, Charlotte?
Yes, I can hear you, but yeah, now we can see the shared screen.
You see the shared screen or not?
Yeah.
Okay.
But we are on the PowerPoint.
Yeah. Okay, so let's go there. You see it?
Yes.
Okay.
So we have contamination, but covered by
Yeah.
plus.
That's good. Okay. So, in fact, you can. The goal of the negative control is just moving alone now. Whatever. The negative control, its goal is to check for contamination. But in fact, somehow you cover already contamination by the positive control and by the alien control. So a question mark, we don't answer it, but a question mark is it really useful? Do we need absolutely to use a negative control? Maybe yes, maybe no. We don't know. We will discuss a little bit about this during the specific workshop tomorrow. So let's go back to the presentation. Here it is. So I show you that for the controls, they are novelties there, and we have also question mark there over there. This alien control is new. The positive control can act as a negative control. The internal control, for example, for viruses are really recommended, or even for bacteria. You analyze the bacterial community, you spike a very little amount of an alien bacteria within it, and to be sure you can detect it. It may be dangerous because if you have a low concentration of bacteria in your sample and you had even a teeny amount, it can represent a significant percentage. So it needs to be tested anyway, but that's an option. And what we did in the guidelines, we listed the control in the guidelines, stating which reference material can be used, for which HTS test, shotgun, applicant, what is the pest range that can be controlled with the reference material? And we added also some notes and remark there. What we did also, we make another graph with these four kind of control, explaining what they are monitoring. Negative control is monitoring contamination. Positive, contamination, analytical.
Sébastien, we cannot hear you anymore. Yeah, I'm sorry, everybody, but Sébastien is offline. So I hope he will reconnect and we will be able to end the webinar. So sorry, Sébastien is still offline, so we will wait just a few minutes to see if he is able to reconnect. I am very sorry about this. So I'm very sorry. I'm trying to contact him another way.
No, I'm really
I
sorry.
will put you as presenter, so because I just.
But on the mobile, I can't. I don't have it.
Yeah.
So, okay, well, let's continue. I'm really sorry. You'll have the slide anyway. And that's now I wanted to thank all the contributors, but well, anyway. The last step and the last point of the guidelines are the reporting of the results. And we take into account three important points for you. This is first, what's happening with inconclusive results. What should I do if I have inconclusive results? I'm not sure about what I have detected. What's happening? How should I report unexpected organisms? Let's think about one of the scenarios, detecting a known pest on a new host. And also we provide information on a remark and also disclaimer that needs to be added with a report. Any diagnostic report is with a disclaimer, and it's important to keep it also for high throughput sequencing. So I go for the conclusion of what I wanted to share with you. In fact, what we have done here is really a comprehensive work to improve the reliability of high throughput sequencing for diagnostic laboratories, but also for research group. What is written in this guideline really aim to help diagnostician and researcher to improve the reliability of high throughput sequencing tests. And in fact, these guidelines, there are 89 pages in total with several chapters. I have them on the slide, but it's not useful to explain it like that. What is really important, and we designed this on purpose, is they are not linked to any technology. I have told it and I remind it, the goal is really to have long-lasting, that these guidelines are long-lasting. It's a worldwide collaboration of 54 experts in the field. They are all in the slide here. You may see it on the PDF. I will not cite them, sorry, if you are among them. But there is one person I really would like to thank. It's Bénédicte Lebas, a postdoc researcher, modern researcher, also a plant risk, plant phytosanitary specialist that coordinated with me the writing of this guideline and allowing all these big group of people to deliver these guidelines, which are really a consensus from all the specialists from the different fields: bacteriologists, virologists, mycologists, nematologists, and entomologists. These guidelines, be aware, they will be publicly available soon. We are adapting them into a scientific publication for peer-reviewed journal in order to really foster the diffusion, not only to diagnostic lab, but also to research laboratory. And finally, yes, I thank you for your attention, your understanding of the technical difficulties, and your participation to the different pools. I will copy-paste the results of today in the presentation that you will receive as a PDF. Thank you for your attention, and feel free to ask questions when needed.
So, Sébastien, there was one question. What would you consider as suitable reference material for validation of the HTS assay?
For the HTS, what?
Test.
A test, okay. In fact, it depends on the scope. So that means the target and the mix. So let's imagine you want to have like using it in for example, what I am doing for indexing banana germplasm here. So what I do, in fact, my reference material corresponds to plants that are infected by the main viruses that we may found on banana. And so my reference, let's say my positive control is in fact a mix of these different plants with the viruses, the most frequent viruses that are present. And the good point is that these viruses, in fact, they are positive single-stranded RNA, there is double-stranded DNA, single-stranded DNA. So we already include with these viruses quite different kind of virus. So this is a good, for example, reference material, because this material has been tested several times, and we are sure they are infected, and the presence of the virus is regular and at a level of infection that is detectable by the test. So that's an example for virus. Let's take another example. If you want to analyze the fungal presence, the presence of fungal pests in a spore trap. In that case, your reference material can be, for example, a mix of fungi that you know you look for. And for example, we just had a recent publication where we wanted to evaluate high throughput sequencing and real-time PCR for fungal forest pathogen. And we used, in fact, 10 species of fungal forest pathogen to make the reference material. And what we did in order to go further, for example, is that, in fact, we do not create a single reference material corresponding to a mix of these fungi, but we do, in fact, 10 different mixes. And in some mixes, we make a very low concentration for one or several species. So in terms of reference material, we had like an equimolar concentration of all the fungi, but we had also sometimes 3, 2, 4 fungal species that were at very low concentration. And so this specific combination allowed us to evaluate if, okay, I can still detect all of them, even if they are diluted 1 on 100, 1 on 1000. So that the reference material is really depending on the target that you want to analyze and your matrix also. I mentioned about spore trap, about banana. Banana, I check on banana viruses. Spore trap, I focus on forest pathogen. But if you are, for example, in a post-quarantine, you have to test quite a wide range of plants. That's a usual situation for some laboratories. In that case, you might need more complex or more reference material, one per species or one per family, depending on the importance of the matrix in the result and the selectivity or not of your high throughput sequencing test. So it's really depending. I try to give some example to illustrate, but it's depending on your scope, so your target and your matrix.
So there is another question on controls. Do you think internal controls would sufficiently compete with low titer viruses for reagents and thus affect sensitivity? And how can we come around this?
This is a very good point because it's indeed a key question. The dosing of your internal control is very important because if it uses all the reagents and all, you will get plenty of reads from your internal control. So for that, what you need, in fact, again, it's validation or verification if it has been tested. The key element there it's also to have standardization of your internal control. It might be good to have, for example, you lyophilize your material, you grind it, and you make already aliquots of it, and so they are properly stored -80 to ensure the stability, and you use the aliquot progressively, taking to try to be always as much reproducible, repeatable as possible with your internal control. Because indeed, if you have, for example, a living plant, and the title is changing, sometimes you will have a lot of reads on your internal control, sometimes not enough. So it can puzzle your results. So this means for your internal control, you need a stable concentration of the virus, either on the plant, it may happen with some viruses, or through stored tissue. But after doing the validation phase, or even the selection development phase, you need to fine tune a little bit which is the proportion that I have to add in my plant tissue to not to avoid false negative of low titer virus. So basically, you compare the same sample with and without the internal control. Do I still detect my viruses that are very low concentration? So it's about, before using it in routine, evaluating it to fix it for the routine. Once you go for the routine, you cannot change it and say, well, sometimes this quantity, sometimes this quantity. You need to find a way to standardize it.
And maybe one last question. What is the impact of mixed infection organisms on the ability of HTS to detect targets?
This is, in fact, if you think about, well, for amplicon, I will make the two different examples. The first, amplicon sequencing. Amplicon sequencing, think about what's happening with quantitative real-time PCR. If you want to be quantitative, we look at the CT once it is the fluorescence is just crossing the threshold. And we know that very late CT, or looking when we go to the plateau phase, we lose the ability to quantify and to have a good overview, because at the end, as the reagents are lacking, there is really a lack of some reagents, the proportion between different targets is not respected. So for this, it's important to first on the for the amplicon sequencing to make low number of cycles, usually 25, 20. And there, you have to keep in mind that what you have with amplicon sequencing, it's relative proportion, not absolute quantification of your target. So even if, for example, you have, let's say, a bacteria which is at 1,000 cells in a sample and 1,000 cells in another sample. If in the first sample you have a heavy load of microbiome, so microbiome rich, let's say it's the soil, you have 1 billion of microorganisms per gram, there you might have difficulty to detect the 1,000 cells of your bacteria. Whereas if you have, for example, you are in the with the endophyte population, you have much less diversity, much less quantity of cells. These 1,000 cells will be easily detected. So that means if you want to evaluate this, you need to make your validation in the conditions that are simulating the routine analysis. And so there you will be able to say, okay, I am able or not in this condition to detect these 1,000 cells that I have spiked in my sample. And another way to cope with this analytical sensitivity and ability to detect low abundance targets is always to compare your high throughput sequencing with PCR, or the reference method being PCR or ELISA, whatever. So that's for amplicon. For shotgun sequencing, there is also another element, is that if you think about viruses in a plant, as I mentioned before, the presence of a virus, and Thierry Candres showed it very well for an Andorna virus. Once this Andorna virus is present, it is, we don't know the mechanism, but it is copying all the resource of the cells and just multiplying at very high amounts. And it is very abundant in the sequencing reads. And the remaining viruses may be harder to detect, and you might need to have a higher sequencing depth to detect the virus species A, whatever it is, if the underlying virus is present. If this underlying virus is not present, this virus A will be much more present in the sample. So again, it brings us back to the representativeness of your reference sample, of your sample panel, for making the validation, the selection, the development of your test. You need to have a view, and you know it well because you're working with this plant, with these viruses. You know already how they behave, more or less. To think, how can I design properly my sample panel to be representative of such cases that may occur, if you know that they may occur quite often. So I try to give some example with the different protocols, but there is plenty of other examples in the literature.
So we have passed the time. So thank you so much, Sébastien, for the presentation. Sorry to everybody that the end of the webinar was a bit chaotic and that Sébastien got disconnected. So thank you for staying online until the end, and thank you for participating to this webinar and also to the series if you were there to the previous webinars. There will be a satisfactory survey at the end of the webinar, and in the follow-up email, it is really short, so do not hesitate to provide your feedback. And as we said, the presentation and the recording of the webinar will be available on the website, so you can double check it if you want. So it was the last webinar of the series, so thank you so much to everybody for following it, and thank you again, Sébastien, for the nice presentations that you gave. There will be a practical training session tomorrow and on Friday for the one that have been registering. We will see you then tomorrow and on Friday. So thank you, everybody, and see you next time. Bye-bye.
Bye-bye. Thank you very much to all.